All right, I think we'll begin. Well, hello everyone. I'd like to begin by welcoming all of you, both students from the University of Washington and the general public, to this very special seminar course called Exploring and Understanding the COVID-19 Pandemic. My name is Jason Beste, and I'm an infectious disease physician at the University of Washington School of Medicine and will be your course instructor. We created this course with the hope of disseminating evidence-based information to you because we're all experiencing the effects of how COVID-19 is not only impacting our daily lives, but also the anxiety and confusion it causes. This is a trying time for all of us, and providing you with the material and information covered in the seminar, we hope that'll help you better understand this pandemic. Since we only have 50 minutes for each session, and that this is our very first session, I will only briefly review the logistics of the syllabus and the course content today, so that you can spend most of our time listening to our two speakers and the Q&A session. If you have some free time after class, we may extend our session by 10 minutes, so feel free to join us until 5 p.m. But if you have to leave at 4:50 p.m., please do, because that's when class is scheduled to answer. As an overview, this seminar consists of 6 sessions, with each session comprised of 2 15-minute lectures followed by a 20-minute Q&A session. The 6 sessions we will cover over the next 6 weeks are coronavirus, emergence and trajectory, which is what we're going to talk about today, pandemic preparedness and response measures, 3, diagnostics and testing, 4, treatment and vaccination development, 5, personal and institutionalized prevention, and 6, the social and economic impacts of COVID-19. Now, these sessions will be facilitated by faculty at the University of Washington who are coronavirus and pandemic preparedness experts. Now, since this seminar is available to both students and the general public, we will be recording these sessions during our class on Monday, and we'll post the webinar for the general public by Thursday afternoons. Although we're using Zoom, we're using a webinar format since we have so many students enrolled in the class. So what this means to you is that when you log into our Zoom sessions, you'll be automatically muted and the chat function will not be available. However, during the 20-minute Q&A session, which again will follow the 2 15-minute lectures, a Q&A function on Zoom will be enabled, and this is where you can type your questions. We'll be answering your questions during the Q&A portion and not during the 15-minute presentations. It is important to note that we may not be able to answer all of your questions since we only have about 20 minutes for the Q&A. Therefore, we do apologize if we're not able to answer your questions. As for the syllabus, we ask you to review it, especially the learning objectives and the assignments section. And if you have any questions, please email Chingling, who is our wonderful TA, and myself. I would like to highlight one section though, which is the assignment section. The assignments for this class consists of 6 quizzes, which will be posted after the session. You will have 1 week to complete the quiz, and the material that's covered on the quizzes will be discussed and presented in the lectures. So as long as you attend the lectures and you listen to the lectures, you should do fine on the quizzes. If you do have any concerns that you may not be able to turn the quiz in on time, please email Chingling and myself. Okay, since our time is short, I'd like to move on to our introductions. I'd like to begin by introducing our wonderful TA, Chingling Hong. Chingling is a 3rd-year MSW and PH concurrent degree student at the University of Washington. His research focuses on reducing the HIV, STI-related disparities among LGBTQ populations and communities of color. Chingling will start his PhD in social welfare this fall. Now, we are very privileged and honored to have our first 2 speakers of this seminar series be Dr. Judy Wasserheit and Dr. Peter Rabinowitz. Now, Dr. Wasserheit has worked extensively at the interface of sexually transmitted infections and HIV clinical epidemiological research, programs and policy in the United States and globally. Currently, Dr. Wasserheit is the professor and chair of the Department of Global Health at the University of Washington, as well as a professor of medicine and epidemiology. She was the founding chief of the United States' NIH's STD research branch, director of the United States CDC's STD and HIV prevention program, and the director of the HIV Vaccine Trials Network. Her development of the concept of epidemiological synergy between HIV infection and other sexually transmitted infections has had a major influence on HIV prevention policy and programs worldwide. Dr. Peter Rabinowitz is an associate professor in the departments of Environmental and Occupational Health Sciences, family medicine, global health, and epidemiology. He directs the UW Center for One Health research that serves as an incubator and organizer of research training and clinical activities related to the human-animal ecosystem interface. Focus areas of the center research include the microbiome sharing between humans, animals, and environments, antimicrobial resistance and diagnosis, treatment, and prevention of zoonotic diseases. And lastly, as I mentioned, my name's Jason Beste, and I'm an infectious disease physician at the University of Washington School of Medicine. And in addition to providing clinical care at UWMC and Harborview Medical Center, I'm involved with medical and graduate education, where I direct the UW School of Medicine's MD-MPH program, the Global Health Pathway, the Global Health Immersion Program, and the UW Global Health and Rural Health Fellowship. My research interests include health system strengthening in conflict and post-conflict settings, and I also work as an international clinical advisor for UW's International Training and Education Center for Health, where I specialize in global health HIV projects. Now, with all that said, I would love to pass it off to you, Dr. Wasserheit, to begin your presentation.
Great. Well, welcome everyone. Jason, thank you so much both for that introduction and for organizing this COVID-19 seminar in record time. This explosive COVID-19 pandemic has radically and probably irrevocably changed the lives of many people around the world, including ending far too many lives. In many ways, this has been pretty close to the nightmare scenario that many of us working in pandemic disease preparedness have long feared and been working hard to plan for. We will ultimately control this pandemic and its health consequences, but I think the economic, social, and political impacts are likely to have pretty long legs and may reshape this era and many aspects of life as we have known it. At the same time, the pace of new insights, innovations, and both the kinds and the quality of collaboration have been unprecedented. So as Jason said, this seminar is going to be an opportunity for us to explore the COVID-19 pandemic together. Today, I'm going to start with a brief overview of the epidemiology, and then I'm going to turn this to Peter to talk briefly about the origins of the pandemic and a little bit about pathogenesis. And we both hope that this will be a springboard for your questions and comments. So with that, I am going to go ahead and start sharing my screen. And hopefully, yes, there we go. All right. So you see here a snapshot of the COVID-19 pandemic. These are the numbers as of today, actually around noon today. But one of the stunning things about this pandemic has been the speed of growth. So these numbers, some of them are actually already out of date. They'll certainly be out of date by this evening or tomorrow. But it's the overarching story that they and the other numbers that I'm going to show you tell that we should be really paying attention to. In less than 4 months since COVID-19 was first recognized, there have been, actually, these numbers are now over 1.9 million cases and more than 100,000 deaths reported. About actually more than a quarter of these cases are here in the United States, and there's a substantial burden in Washington and right here in King County. And we're seeing case fatality ratios in the 4 to 6, 6.5 range. But it's actually very hard to interpret these numbers because there is substantial underdetection of mild or asymptomatic cases, which we know are probably at least 80% of infections in this pandemic. And there's also probably, to a lesser extent, underreporting of COVID-19 related deaths. So in fact, we believe the true case fatality ratio is probably going to turn out to be closer to 1 or less than 1. We'll come back to that in a little bit. That snapshot really reflects a collage of epidemics around the world. And I'm showing you here curves for 9 countries with the daily counts in red and the daily case counts in red and the daily death counts in gray. And the different shapes that you see on this slide, the different curves, are due to multiple factors, including differences in age structures, urban-rural splits, travel patterns, healthcare system capacity, and the timing, type, and adherence to social distancing and other policy interventions. And we're seeing equal diversity of epidemics, localized epidemics across the United States. Here in Washington, the cases women are slightly, but not extremely overrepresented in the number of reported cases or the proportion of reported cases. But it's striking both here and across the country and in other parts of the world that men clearly predominate among deaths. While cases peak in young adults and middle-aged adults, as one might expect, because those are the people who tend to be more out in the world, less able to social distance in some ways. Severe disease resulting in deaths disproportionately affects those who are 60 years old and older than that. And again, this is not just in Washington. We're seeing this across the country and around the world. Here in King County, we're also seeing the emergence of racial and ethnic disparities, which is also being reflected in other parts of the United States, with members of marginalized communities of color being disproportionately affected. And this too is not surprising because these are people who are more likely not only to have more limited access to information, but more limited ability to social distance. Although snapshots are very valuable, what we really want to look at is trends over time, because these tell us where we're going, which is what we really want to know. Since the end of March, as you can see in this upper curve, for the state of Washington, we're beginning to see a decline in the number of new cases reported. And as we would expect, this is beginning to be followed by a decline in the number of deaths reported, which is shown here in the orange curve below. But obviously the deaths follow cases with a lag. This is all in the context of robust increases in testing since the middle of March, with even greater numbers anticipated in the coming weeks. And these curves that you see here are peaks during the weekdays with declines over the weekends. These are weekly trends. And We are also beginning to see the possibility that hospitalizations may have begun to plateau, which is also encouraging here in Washington. So all of that brings us to that mantra that we all keep hearing, flatten the curve. The concept that we need to implement policies and non-pharmaceutical interventions to buy time so that we don't overwhelm the healthcare system capacity and therefore are able to save lives by continuing to deliver high quality care. The other thing that we're trying to buy time for actually is to give researchers a chance to the kinds of, to develop and test actually, the kinds of tools that we need to deal with this pandemic, diagnostics, drugs, and vaccines. Another way to think about flattening the curve is that we actually want to increase the doubling time of national and localized epidemics. And hopefully this will work here. Good. This is encouraging. Yes. Let's see if we can do this. I think this is a beautiful depiction of how these
Judy,
slopes of these epidemics have changed around the world.
I'm sorry to interrupt you, but we actually can't see that picture, what you're showing.
You can't see
Nope.
this.
I think you have to hit share screen again, or end your screen, and then share screen again.
Actually, yeah, Judy, you want to share your whole desktop to see that and then go back to the PowerPoint after you're done.
Okay, talk me through this. Hang on a second here. Let me stop this. Stop it. All right. So you're telling me I need to go out of this?
Yeah, now you can either share your entire desktop, or if you wanted to share the internet browser, you can choose to share Chrome, wherever you're in, whatever browser your information is showing up on.
Okay, let's see if I can do that. This is still not going to work, I think. Let's see here. Stop share. See if it will let me.
And then now end the share on the PowerPoint. What do you see for options for when you click on share screen?
Let's try this. Can you see that?
Yes, that's it.
Let's try this. All right. So what is depicted here are slopes for this curve. And watch what happens here. If I can, yeah, here we go. You can see China coming. No, now it won't. Well, this is the challenge of Zoom, folks. I apologize. I'm going to go out of this, and what I was hoping to show you was how all of these curves progressed. But let me just do this a little differently. Apologies. Can you see my screen now?
Not yet.
Okay. Let's try this. Can you see my screen now?
Yes, we can.
Wonderful. Okay. Well, I apologize for not being able to make that video work on Zoom more effectively. But the endpoint of that video would have shown you this, this figure. And you can see, for example, for the United States, how the doubling rate declined from roughly a doubling rate of every 2 to 3 days to now about every 9 or 10 days for the United States. And here you see the same or an analogous set of estimates for major metro areas, with Seattle having now moved from decline to about a double time of every 2 weeks, which really gets us to the point where we start to be beginning to be able to control this epidemic right here. Again, I want to emphasize, however, that there is probably substantial underdetection of cases. So people have raised questions about the reliability of some of these estimates. But overall, I think the message is that we are beginning to see traction with some of the policy interventions in some cities and some parts of the world. So the real question, of course, is what do we do to flatten the curve and ultimately control this pandemic? One thing that I learned as an infectious disease physician and epidemiologist is that if you want to beat a virus, you actually have to think like a virus. So if I were a virus, I would most of all want to do 2 things. I'd want to survive as long as possible, and I'd want to reproduce as much as possible. One of the best ways to spread and both survive and reproduce is to link my spread to essential human functions. And 2 of the best ones are breathing and sex. This virus, of course, is a respiratory virus, and the primary transmission mode is large droplets as a result of coughing or sneezing. Second thing is to travel undercover. We do know that this virus is transmitted even in the setting of asymptomatic, minimally symptomatic, or pre-symptomatic cases. Although I have to say we don't fully understand how efficient those different types of transmission are. The third thing is we want to, as a virus, I would want to stay as infectious as long as possible. We're still learning exactly what the infectious period is, but it's pretty clearly longer than the symptomatic period. The fourth thing is that I want to be slow to kill or severely debilitate my host, so that I could have a way to be carried to the next, to infect the next person. And with more than 80% of cases being minimally or moderately affected, I think I'm in good shape here. Finally, I want to be able to change my appearance, change my coat. And here we're still learning exactly what kind of mutation or evolution rate SARS-CoV-2 has. Which brings us to the concept of the reproductive rate, or R sub 0, of infection, the average number of new infections resulting from each index infection. The reproductive rate is the product of 3 factors: beta, the transmission efficiency; C, the contact or exposure rate; and D, the duration of infectiousness. So to control and ultimately end a pandemic, we need to drive R sub 0, or the reproductive rate, down to less than 1. So one way to do that is to decrease C, the contact or exposure rate. And that's what we do with social distancing, things like hand washing, masks. But if we want to be able to drop back and de-escalate from social distancing, let our contacts with other people start to increase, the other way to do that is through vaccines or treatment, which decrease beta, the transmission efficiency, or the duration of infectiousness. So what do we actually know about this virus that causes COVID-19, and how does that compare with viruses that cause SARS and flu? Well, first of all, we know that the reproductive rate for COVID-19 or for SARS-CoV-2 is similar to that for the virus that caused SARS or causes SARS, as are many of the other key parameters that I'm showing you here, except that the virus that causes COVID-19 seems to result in more asymptomatic transmission. And also the case fatality ratio is actually lower than that for SARS. It is noteworthy that the reproductive rate for COVID-19 is greater than that for seasonal flu, although it's about the same as for pandemic flu. And the case fatality rate sits somewhere between that for seasonal flu, for which we do have biomedical interventions, and pandemic flu. So finally, what do we need to do to end this pandemic and de-escalate? Probably at least 8 things. First of all, we're going to need widespread viral and serologic testing, ideally home-based, not just home-based specimen collection, but actual home-based testing, sort of like pregnancy tests. And that'll have to be linked to more extensive contact tracing, potentially even assisted by mHealth platforms or IT assisted, and there's some work beginning on that front. We're also, of course, going to need both therapeutic and prophylactic drugs and vaccines, but then we need to have the delivery systems to get those things to the people who need them, and the surveillance systems to be able to do the monitoring and evaluation to understand when we need to expand back to more social distancing for a time and then back off. We also need sustained funding for pandemic disease preparedness programs and research, something that we have never had, despite a series of pandemic outbreaks previously. And finally, we are going to need, these pandemics by definition are global. So we need ongoing global collaboration for prevention and control. And with that, I'm going to turn this over to you, Peter. I'm going to stop sharing my screen, and the Zoom is yours.
I have to make sure you can hear me. Thanks a lot, Judy. And in the interest of time, I'm going to jump ahead to taking a slightly different look at the COVID-19 pandemic, because just as we are so engaged with trying to respond to the current pandemic and trying to flatten the curve, trying to look ahead to real solutions for this particular pandemic in terms of vaccines and therapeutics and diagnostics, I think it's worth spending some time just thinking about why this happened in the first place. What was the origin? What do we need to know about that so that we can prevent future pandemics like this? Because there are a number of other threats like this waiting to come down the pike, and it's not enough to just deal with this particular pandemic. We have to realize that we're in an era where these pandemics are coming more frequently, and we have to be able to do something about that to prevent them at the source. So just to go over some basic definitions, when we talk about pathogens, we're talking about a microbe. It could be several different times, viruses, bacteria, capable of causing disease. There's this whole question about why infectious diseases are emerging more than ever before. And this is basically emerging infectious disease is a new pathogen that we've never seen before, like the virus that causes COVID-19, or a previously known pathogen that's changing its range or behavior, sort of like influenza can change and become a new strain that is all of a sudden emerging. I want to talk about the issue of zoonotic disease or zoonoses, and these are diseases transmitted between animals and humans. And COVID-19 is an example of that kind of disease. And we then have to think about the fact that there are many animals that we share the earth with that are carrying around pathogens that are capable of causing disease in humans, causing zoonotic diseases that can turn into pandemics just like COVID-19. And when you count up all the different human pathogens, there are people who've made a catalog of every pathogen that's been discovered, and you say there's about 1,400 different types of viruses, bacteria, fungi, protozoa that cause disease. 58% of them, 816, are zoonotic in origin. So there's basically animal diseases that have now jumped over to humans and are causing disease in humans. And when you look at the 177 emerging pathogens in the recent 2 to 3 decades, 73% of them are zoonotic. So there's really something going on here about these new emerging infectious diseases that relate to diseases coming from animals to people. And we don't have to look far for examples such as Ebola virus, HIV, avian influenza, and now COVID-19. So want to really enforce the fact that this SARS coronavirus 2 virus that causes COVID-19 is an emerging zoonotic pathogen that we've never seen before. It's a novel pathogen, and it comes from animals. It's a RNA virus. It tends to attack the respiratory system. And as Judy was saying, if you can be a virus that attacks something as important as breathing, that's a good thing if you're a virus. There are many types of coronaviruses, both in animals and in humans. So there's a number of coronaviruses that are somewhat unrelated to SARS coronavirus 2, and they cause very mild cold-like symptoms every year. When we track that, we've known about that for a long time. But some of these strains that are zoonotic and moving from animals to people seem to be much more virulent. And here's a picture of what these coronaviruses look like. They're called coronaviruses because they sort of have a corona or a crown of these spike-like proteins that go around them. And these are RNA viruses, and RNA viruses in general are able to mutate fairly quickly and behave in different ways. And Judy mentioned SARS that you hear about sometimes, the original SARS. And what was that? That was a coronavirus that emerged in 2003, 2004 in China. And it was caused by SARS coronavirus 1, and it caused severe acute upper respiratory syndrome. As Judy said, it was more higher case fatality rate, more virulent than SARS coronavirus 2, but it really had not nearly as many people affected. Less than 10,000 people were infected. And as a result, it just didn't spread as far and kill as many people as SARS coronavirus 2 has. And that was the first time we had seen a novel coronavirus emerge from animals like this in sort of recent history. But not that long after that, about 10 years later, in the Middle East, all of a sudden we found another emerging coronavirus, SARS, which was MERS coronavirus, Middle Eastern Respiratory Syndrome. This turned out to be transmitted from camels to humans, but we think that both SARS and MERS have their reservoir in bats that are carrying it around the world and then giving it to another animal that then gives it to people. And if you think about how zoonotic diseases work, this is a virus in the middle, and it has a natural host that is usually asymptomatic. So for many diseases, such as SARS, such as MERS, the natural reservoir is bats. For influenza, the natural reservoir is geese and ducks. They don't, and so bats and geese and ducks don't get sick from these. But when MERS went into camels, the camels actually didn't get that sick, but then they were able to infect humans. They then with SARS, we think that the virus went from bats into civets, a cat-like animal, and then into people. So there's these intermediate hosts that sometimes can actually get sick, but are very important in sort of genetically allowing the virus to jump to people. And this is an issue that you may have heard about, the term spillover, where we think, as we think about all the wildlife in the world, all the animals in the world, there are little amounts of pathogens that are increasing or decreasing in frequency in an animal population, in this case, a wildlife population. And then at times they can spill over into susceptible populations of both domestic animals as well as humans. And these sort of crowded, susceptible populations can amplify the virus and turn a very small little fluctuation in pathogen load in an area into a large epidemic or pandemic. And that's the issue of spillover and amplification. So what happened with SARS coronavirus 2? Basically, we don't know exactly the origin, and we need to know. We know that around December 2019, there started to be cases in Wuhan, China, and many of the early cases, but not all of them, were associated with seafood animal markets where animals were being sold. And when we look at the genomics of the isolates of this virus, this large number of viruses shows that we're sequencing lots and lots of these viruses, and they're showing little differentiations. As Judy said, we're still not sure whether small changes in the genome of the virus give it any really different qualities in terms of transmissibility or virulence. But we see that in general, that there is a close relationship between these SARS coronavirus 2 isolates and some bat coronavirus isolates that we think were the ancestor of this SARS coronavirus 2, but to get this little genetic evolutionary change from the bat coronavirus to the SARS coronavirus 2, we think involved another animal. We don't know what that animal is. In the same way, this is SARS, and SARS is also very closely related, well over 96, 97% to bat coronavirus, as is MERS. So we think these are all related, but we think that there's often an intermediate host between the bats and the human infection. So this gives us some reassurance that this is not an engineered virus. All the rumors that you might have heard, the genetics argue strongly that this has jumped from animals to people, just as SARS and MERS did. And we think about the seafood markets in Wuhan. These are a picture of the markets when they're open, and then they got closed after the outbreak. What was going on in there? What kind of wildlife were being sold there? We've heard different reports, but we've actually have not seen any scientific study of it. We know that there's a large wildlife trade in many parts of the world, and China is a place where there's a lot of wildlife trade, both for food and medicines, but we don't know whether it was wildlife that did it. And we wonder, is there an animal like the civet that was the intermediate host for SARS and was present in the markets and got infected somehow and then spread it to other animals in the market and spread it then to people? What's the missing link like that? So somehow we want to know, how did it go from the bats into some other animals? And there's been different candidate animals. Pangolin, there's a question mark here, because even though there was some genetic similarity, we haven't seen the evidence. So what is it that is allowing a natural bat virus to go to another animal and then go to people with SARS coronavirus 2? Well, the evidence is that of the original 41 patients that had contact in the Wuhan seafood market, and we put this paper in the recommended reading, 27 of the original 41 patients did work in the market or had contact with the market, but the very first case that was sick around the 1st of December did not seem to have any connection to the market. So we don't have a clear smoking gun there that we'd like to see. And right from the very beginning, there was person-to-person transmission, even in these early cases. At the same time, they did report some samples from the market, and a lot of the positive ones came from the part of the market where there are animals. So we think there's tantalizing evidence here that something happened, and we think that was probably where some original spillover happened. But alternatively, the spillover event could have happened somewhere else, and then an infected person or infected animal went to the market, infected people there. So it's not totally clear that it all happened in the market. And partly the problem is that we have not seen research published. We're not sure why, but there's no study of the workers of the animals in the market published to date. We also have to think about a little bit of biology about the virus. We see that it uses what's called the ACE2 receptor, as angiotensin 2 converting enzyme receptor, and it's a membrane protein which basically SARS coronavirus 2 uses for cell entry. It uses it to get into cells by binding to it with the spike proteins. This receptor is present in human lung, heart, kidney, and intestine, and we see that the SARS coronavirus 2 is much better at binding to it, to our receptors of ACE2 than SARS coronavirus 1. And that could be why SARS is spreading more than the original SARS coronavirus did 20 years ago. What it did, I'm just going to set up a little bit the later lectures about the virus. The pathophysiology is that most cases are mild. It does tend to go to the lung and cause inflammation and pneumonia, and you die from respiratory failure with ARDS. And in some cases, we think the immune system, our own immune system that's supposed to be fighting the virus, actually gets so overactivated that it destroys our own tissue, and we have what's called a cytokine storm, which is just, basically we're damaging our own tissue by having too much immune reaction. So those are things to think about in subsequent parts of this course. But what I want you to really leave you with is the bottom line is that we have to remember that there are many other coronaviruses and other pathogens out there in natural animal reservoirs. We cannot go and get rid of all the animals on the planet. That's a terrible idea. It won't work. It won't work ecologically. So we are going to have to coexist with animals and wildlife, and we want to do that anyway on this planet. But that just as we are social distancing from each other right now to flatten the curve and control COVID-19, we need to think about, as a society, a global society, we need to maintain some sort of distance between the human populations and the animal pathogens. So we need to create a social distance between us and the bats, or there's going to be more spillover. And there's something called the One Health approach, where you think about how to make that kind of distance, how to coexist, thinking about the health of the humans, animals, and environments all at the same time. So for discussion, just think about, what actually happened, what could have been the animal host, what's the role of the market, but really how do we work together to prevent the next SARS? We need to do something about our way we relate to wildlife and the wildlife trade, and how we coexist on this earth with animals that have pathogens that we would rather not share. And I will stop there.
Great. Peter and Judy, thank you so much for those presentations. Wish you could hear the distant clap for all of your great work and your presentations that we're all giving to you. We do have a bunch of questions that have come up, so thank you for all of those who have typed in questions. Keep the questions coming in that Q&A box, so if you just click on that, you can type your questions. So let's get started. So maybe I'll just actually start with you, Peter, since you just finished your lecture. There's a question that came to you that asked, we've had 3 major coronaviruses emerge in the last 20 years, SARS, MERS, and now COVID. Is this due to something that's unique to this time period? Or is this a phenomenon that's been happening for a long time and now we're just able to monitor it?
What we think is that it's unique to this time period. We think that as never before, the human population is having interaction with wildlife and then having situations where that interaction can turn into a real disease event. And some of the things we're doing is we're destroying habitat, so the wildlife are living on smaller and smaller bits of habitat. We're living up close to where the wildlife are, so we're moving our communities in close proximity. Sometimes the wildlife actually like to come into cities and live right in with us. We have more crowding of people, more contact with wildlife, and then creating these sort of susceptible populations of both people and domestic animals. So like lots of pigs all together, all susceptible to a new virus, lots of chickens all together, all susceptible, lots of people all together living in a city, fairly susceptible to a new pathogen that comes apart. So again, thinking like a virus, they think this is great. I've been with this animal for a while, but I can jump into a whole new population very easily and spread around it. And that's what these ones are doing. So we've seen more emerging infectious diseases in the last several decades than we think we've seen in hundreds of years altogether.
Thanks, Peter. Judy, a question to you. You gave us a lot of good definitions around epidemiology, and one of the topics you talked about was the doubling rate, and one of the students asked, what is a doubling rate?
That's a great question, and I apologize for not explaining it initially. It's the time that it takes for the number of cases to double in a population.
Great, thank you. And maybe to the both of you, is it known why COVID-19 was able to spread on a global level compared to, say, MERS or SARS? Or why did it spread a lot faster and more efficiently?
So we're still understanding the biology. We're still, and we need to understand it quickly because we're trying to make new vaccines and drugs that attack the way it's working. But we do think that it's very good at binding to these receptors and then causing enough viral load in the body that allows us to put out lots of virus as well. And it looks like we can put it out from our lungs and cough it out, but that we do seem to shed it in our stool for a while. And so it tends to be something that we're pretty good at being spreaders of, as well as it's good at binding to us, I think.
I would say they're, at the biological level, what Peter just talked about is really important. At the epidemiologic level, two of the differences are that, number one, there seems to be more asymptomatic transmission, and that's really hard for us to deal with, because if somebody doesn't have symptoms or has really minimal symptoms and doesn't present for care and goes about their daily activities, that's a great way to spread. And the other thing is, you may remember I said that the case fatality ratio is lower for COVID-19 than it was for SARS. And I didn't show it to you for MERS, but where it was about 10 to 15 for SARS, it was about 36 for MERS. So when we're down in the 1 or lower range, that's a lot less. And, you actually don't want to really kill your host fast, because then you can't spread. You want your host to survive and be well enough to go help you move. It's like, a taxi that has all its wheels on or not.
And I totally agree. And just to say, one other thing about the difference between SARS and COVID-19 is, and SARS really did sort of die out and has really not come back, which is great. It might, but it hasn't yet. But back in 2003, it's hard to remember that even less than 20 years ago, there was not nearly as much air travel, people going back and forth to China where the epidemic was happening as there is now. China in 2003 was only 4% of the world's GDP. There was just not a lot of trade, and now it's basically 20%. And so the number of people that were moving in and out of countries, coming back and forth to China where it really started, was such a powerful way to seed the virus in many other countries very quickly, which really wasn't happening even 20 years ago.
I do think in relation to that travel point, which is tremendously important when we think about pandemic disease prevention going into the future, we got to recognize that the data now suggests that there are probably at least 8 different clades or flavors of this virus. And it seems like there were introductions, for example, to the United States, both from China, primarily and other countries in Asia from the West Coast, but a lot from Europe, actually, on the East Coast. And as we think about what's emerging in sub-Saharan Africa, where I think this pandemic will really be devastating because of the limited healthcare capacity and for a number of other reasons. I think probably what we're going to see is, again, a mixture of travel, strains that reflect travel from Asia and from Europe. So travel has increased dramatically all over the world.
Kind of a follow-up question with One Health, since we're kind of on this topic a little bit. Is it possible to evaluate viruses while they're still in animals and assess how severe they would be in humans if they spilled over?
We can definitely, so there's been a project that went to look at sort of bats and non-human primates and rodents, all sort of these high-risk animals around the world, and they found hundreds of new viruses that we've never discovered before. So we know that there's many more viruses out there, and we've discovered a lot of them. We need to try to understand them better. We need to be ready to have kind of response platforms that we could detect new viruses very quickly and create vaccines and things for them quickly, knowing something about what's out there. But we have to assume that there's a lot out there, and we have to find ways, again, to just prevent the spillover, because we know pretty well that there are a lot out there. And we can also learn one other thing, which is that the animals that have it asymptomatically, they have natural defenses against the virus, and they could be also a source of scientific information about how does their immune system keep the virus in check when ours can't. What can we learn about that in terms of therapy or vaccines at the same time?
Great. And I just want to take a quick pause just for everybody, all the students who are on the line. It is 4:50, so I do know it's time. If you have to go, please go. If you are able to stay, we're going to try to go for another 10 minutes or so, because we have a lot more questions. But with that, I'll keep going with some of the questions. Here's a question. Are you worried that there will be another wave of COVID-19 once we flatten the curve and social distancing ends?
Well, maybe I'll start with that, and then Peter, you can chime in. I think that many of us believe that it is very likely that there will be echo waves. And we say that for a couple of reasons. One is that we are in uncharted territory, both within countries and around the world. We have never implemented social distancing and other non-pharmaceutical interventions at the scale that we've tried it now. And so we really don't know, we have no experience with de-escalating. And we're going to be learning as we fly this plane, right? I think most people believe that it will be critically important to set certain thresholds that would be triggers for signaling that we might want to slightly upregulate our non-pharmaceutical interventions. At the same time that we increase any biomedical availability of any biomedical interventions that we have by that point. I think people are most worried, including myself, about what will happen in the time frame between when we start to back off on these non-pharmaceutical policy interventions, and we have an ability to deliver not only drugs, but vaccines in a widespread way. And I think pretty clearly, we're going to need to use pharmaceutical interventions like both therapeutic and prophylactic drugs as the interim step until we get a vaccine, because we will have data from the many treatment trials that are in progress right now before we have completed vaccine trials and been able to scale up manufacturing of vaccines.
And
Peter, what do you want to add? What do you think?
No, I agree. The problem is that until we have a vaccine, and there's lots and lots of people that are out there that have not got infected that are still susceptible, it's like a fire. It's going to be hard to really put out until we can have enough immunity around it that the virus just can't spread. And we by relaxing, we've flattened the curve. We've done a lot, but by relaxing, we're just opening the gates a bit, and there's no reason why the virus is just going to die out at that point. So we have to really continue to watch this really closely.
And this
I
question,
think the good news is that we do have, trials of some of the drugs looking not only at treatment, but also at post-exposure prophylaxis. One of them is starting here in Seattle. And people are already thinking about what kinds of increases in hospitalizations might trigger a return to more of the policy interventions of social distancing.
and we're going to have a lecture on vaccinations and treatments and everything what we've been talking about. So that's a great question. And I will say too, at the hospital level, we just had a call on Friday, a big meeting about the same question. The hospital, we've hit our peak, we've hit the plateau right now. So we don't expect to see hopefully more influx of cases. We're very busy at the hospital, but that question came up is once these restrictions relax, what's going to happen to the hospitals and how long will that take to see a second wave? Because as we've been talking, we don't know what the herd immunity is in the community at all. So it's a very concerning question. Speaking real quick about vaccines, we are going to talk about this in a couple of weeks, but one question is, knowing that SARS turned on the alarms and MERS as well, why did we not continue researching for a vaccine for these novel coronaviruses?
I love that question.
Go ahead, Judy.
And I did not plant that, but that is a great question. And the answer is maps against one of my parting messages, which is right now we are very focused on this COVID-19 pandemic and controlling it. But this will not, I promise you, this will not be the last infectious disease with pandemic potential to emerge. And what happened back in 2003, 2004 was that once SARS settled down and it was no longer a problem, funding to continue research on diagnostics, therapeutics, and vaccines went away. And we moved on to focus on other things. And in fact, as Peter and I have tried to develop the University of Washington Meta Center on Pandemic Disease Preparedness and Global Health Security, which is sponsoring this seminar in some sense, it has been really challenging over the last 5 or 6 years, given all the other priorities in health and research, to get people to focus on the idea of preventing something that hasn't yet happened but could be terrible. And so one of my prayers for this is that we will come out the other end and be able to sustain funding for the kind of research that would have delivered vaccines and drugs from SARS to prevent what happened now, but do it much more broadly.
Great. Maybe we'll try to do 1 or 2 more questions. This might be a quicker question for either of you. Do you think COVID-19 will be a seasonal virus like the flu?
You're muted, Peter.
Thank you. So it would be great if the sun came out in Seattle and burned away all the virus. It has not shown strong patterns like that, and it has broken out in many places where it's very hot, and places like Singapore, and so we are not too hopeful. It would be a wonderful thing if summer weather put a dent in it, and that what flu virus does is it sort of migrates to the southern hemisphere during our summer, and then comes back for our winter, and so spends time really preferring the winter months. So far, it's not totally clear that it's going to do that, and I would love to see it happen, but I have not seen compelling evidence that it's that seasonal. So that's where it is for me right now.
Great, thanks. And maybe one more question we'll do. You know, Judy, a couple students have asked a question on one of your slides, and you just got to pull it up here. Here it is. So, question for Dr. Wasserheit. Looking back at your slide indicating 8 things that need to happen to end the pandemic, were you saying that all of those realistically need to happen before this can end, or are some of those more weighted and heavier than others? And I would just add to that, if there's, you know, 1 or 2 things we can really focus on now going forward where we are currently, like where are those priority areas? You got 1 minute.
Right. So, you know, we always want magic bullets. And if you think about the HIV pandemic, which in some ways has lots of lessons to inform what we do here, we finally realized that we were never going to be able to deal with HIV with a single magic bullet. Maybe someday we will have the perfect vaccine, but even then, people have to use the vaccine. There are some behaviors that need to change, right? And so my feeling is that we need a combination of strategies. What these 8 things really say are that we need a combination of the biomedical tools to be scaled up and the systems through which they can be delivered in combination with the kind of surveillance systems so that we can know what's really happening with this disease. And that's a tall order. If I had to prioritize things, you know, I think if you go back to the 3 components of the reproductive rate, if we want to back off on the social distancing interventions, we really need those biomedical interventions to do that. And that's why the research that's going on in those areas and that you're going to be hearing about in some of the later sessions in this, this seminar are so important. But if we have those without the delivery systems and without ongoing support for research and programs to prevent future pandemics, we're going to be, you know, it's going to be like Groundhog Day. In 20 years or 10 years, we're going to be saying, why didn't they do more with COVID-19? And why are we back here, right? So.
Great. Thank you. Well, I'd like to say a special thank you again, Dr. Wasserheit and Dr. Rabinowitz, for joining us today for your great presentations. Thank you so much. For all the students, thank you for great questions. Apologies, we weren't able to get to all of them. A lot of the questions did have to do around tests and vaccinations, and so we're going to have a session on that, so you can ask those questions at that time too. And just remind you, next week at 4 p.m., our second session is going to be on pandemic preparedness and response measures, so it'll be very exciting, great lecture. And your quiz will be posted probably later tonight or early tomorrow, and remember, you have about a week to do that. Again, thank you so much for joining us, and everybody have a good evening and a safe week.
Thanks, everyone. Thank you, Jason Chingling.
Thank you.
All
Bye-bye.
right.
Thanks. Take care.